So this is my first year engaging with Target ALS. It's the first funded project together with Paul Greer and Bob Brown at UMass Chan Medical School. I'm actually a little bit outside the field of ALS research, so it's really nice to dip my toe in the water and see the field and feel it out. Like I said, I'm learning a lot about the research that's going on now, and I'm excited to interact more going forward. So my colleague Paul Greer had identified these MS4A genes in the olfactory bulb. He actually worked in the olfactory system and sensory processing. And it became really intriguing because this became a identified risk gene in neurodegeneration, particularly Alzheimer's disease. So we started looking at this, and it actually seems it's highly enriched in these immune cells that live in our brain and spinal cords called microglia. And so we began studying these cell types in the context of microglia and neuroinflammation, starting with Alzheimer's disease, but now moving into ALS. And it seems like these microglia that are high for this gene are increased, particularly in ALS models, including the SOD-G93A mouse model. So now we're looking at, my lab looking at using these spatial transcriptomic approaches to map gene expression across spinal cords and ask how these MS4A high microglia might be different than surrounding microglia, how they affect their neighbors, and then ultimately identify putative therapeutic targets that we could target to prevent, for example, neuroinflammation and the spread of disease in ALS.